The overall goals of this liver digestion protocol are to assess the various liver progenitor subsets by flow cytometry, and to isolate these cells to a high purity using a novel surface marker cocktail for further downstream analysis.
This method can help answer various questions about the biology of liver progenitor cells. For example, what are their subset-specific features and what are their distinct roles during liver injury.
The main advantage of this technique is that it allows the isolation of progenitor cells with high purity and viability, which is important for their in vitro analysis.
Though this digestion technique provides insight into progenitor cell biology, it can also be utilized for the isolation of hematopoietic and endothelial cell populations.
To prepare a single-cell preparation from a mouse liver, transfer the lobes onto a dry petri dish and use a scalpel to cut the tissues into approximately 2 mm cubed homogeneous cubes. Transfer the pieces into two 15 ml conical centrifuge tubes per liver, containing 2.5 ml of digestion buffer each, and place the samples in a 37 degrees Celsius water bath with gentle shaking at 5 and 10 minutes. After 15 minutes of digestion, use a 1,000 microliter pipette with a cut tip to gently triturate the liver pieces. Then return the tubes to the bath and allow the pieces to settle for 2 minutes. Next, filter 2 approximately 700 microliter aliquots of the supernatant through a 100 micron strainer pre-wet with 800 microliters of collection buffer, and replace the removed supernatant with fresh digestion buffer.